Lip lipids have high solubility in non-polar solvents, an example of which would be benzene, and they have low solubility in water because water is polar, as seen in the picture here. So because of the angle in water, the part with the hydrogens is a bit positive while the oxygen is more negative. So that's polar, and lipids don't dissolve well in water. So some examples of lipids would be fats, oil, waxes, and some of their functions include being a major part of biological membranes, like the cell membrane. They're a good form of stored energy. Burning fats gives us twice as much more energy than burning carbohydrates or proteins. They can be used as hormones to send signals between cells, and they are used as lung surfactants. This is a classification of some of the lipids. They can be simple, complex steroids, and then eicosanoids, which are, they have a carbon chain that contains 20 carbon atoms. This is another way to classify, and this is the one we're going to be looking at more in depth. So lipids can be simple, complex, and then derived can come from simple or complex by hydrolysis. So in simple, we have wax, which everyone knows, like kind of like the wax used to make candles, etc. Then we have sterol esters. This is a picture of a sterol. So instead of the hydroxide here, it has a fatty acid connected. So that would be what a sterol ester is. And then we have triacyl glycerol. So it has a glycerol backbone, and then it's connected to 3 fatty acids. That's just a normal fat or triglyceride that we know. Then in complex, we have phospholipids. So they have this glycerol backbone, and then they also have a phosphate group attached. And then there's 3 types: phosphatidylcholine, phosphatidylethanolamine, or phosphatidylinositol. Or we can have sphingolipids. So sphingolipids, instead of this glycerol, they have something called sphingosine. We'll look at it in more detail later. And then glycolipids are lipids plus a carbohydrate part. For the derived, we have fatty acids. So from here, we said this is a glycerol and these are the individual fatty acids. So if we break it down, take 1 fatty acid, that would be what this is. Sterols. So if we break down the sterol ester from the fatty acid that was connected here, we just get this, which is a sterol. We can also have diacylglyceride or monoacylglyceride. So they come from triacylglycerols. If I remove 1 fatty acid, I'm left with 2. That would be diacylglycerol. And if I remove 2, only 1 fatty acid would be left. So that is a monoacylglyceride. Now for waxes, there are simple lipids, like we said, and then there are an ester. An ester is formed when an organic acid, which is this, it has this C double bond O and connected to an OH group, reacts with an alcohol. So this is the alcohol, it has the OH part. And then when they react together, they form this bond, which is the C double bond with an O. That bond is called an ester bond. And this substance, by reacting a fatty acid with a long chain alcohol, is what an ester is. And they can be found as coating on leaves to prevent water loss. Then we go to phospholipids. So from the name, they are lipids that have a phosphate part. They are a major component of the cell membrane, and they have a hydrophobic tail. So 2 hydrophobic tails, which are these as seen here. So those are the fatty acids, and a hydrophilic head. The head is hydrophilic because it has the phosphate part. So they can be used on the cell membrane as messengers or as anchors for proteins to attach to them. Or if they're not in the membrane, they could be used as lung surfactant, and they're also an important part of bile. So phospholipids are esters. When an alcohol and a carboxylic acid react together. We already said that's how an ester is formed. And this is again the ester bond that I mentioned. A phosphodiester bond is when you have a phosphate in the middle, and then it's connected by an ester bond to two other molecules on two sides, this and this. So this is why this is a phosphodiester bond, and it's found in DNA and RNA. Now, like we mentioned this before too, so it can have a glycerol part connected to two fatty acids and one phosphate. That would be phosphosylglycerol. And the sphingomyelin has sphingosine instead of this glycerol as the backbone. And then it can be connected to a phosphate. So they're just saying it again. So in a phosphoglyceride, the carbon 1 and 2 are connected to fatty acids, and then carbon 3 is connected to phosphate. So if you just have this with no further addition of an alcohol, that molecule is called a phosphatidate. We're going to see the structure again, but a phosphatidate is the simplest phosphoglyceride. So it's just this part without an extra alcohol connected to the phosphate. Phosphosylglycerols again, and they're just saying we have a phosphodiester bond because one bond here with the glycerol and one bond with the alcohol on the other side. That's why it's a phosphodiester bond in the phospholipid. Again, they're just showing how it's arranged in a cell membrane because we said that the head is hydrophilic. So and generally all the environment in our body consists mostly of water, and only the hydrophilic part can face water. So it forms this lipid bilayer where the hydrophilic heads are facing outside. So we have one phospholipid here and the other one in the opposite orientation. So on both sides, the hydrophilic heads will be facing water, while the hydrophobic tails are kind of hidden inside. Now we're talking about phosphatidate again. Like we said, that was the simplest phosphoglyceride. So it has a glycerol and then the fatty acid groups connected to carbon 1 and 2, and then you have the phosphate here. So that's what phosphatidate is. Then we have something called phosphatidic acid. So this is the structure here. And we can break down phosphatidic acid to get one molecule of glycerol and two fatty acids and phosphoric acid. And they make up the phospholipid bilayer in the cell membrane. And they constitute 0.25% of it. Now, these are just other examples of phosphoglycerides. I don't think it's that necessary to know the structures themselves, but we can just go through the names. So this is phosphatidylserine because the group connected to the phosphate is serine, and then here it's choline, that's what phosphatidylcholine. And then we also have phosphatidylethynylamine and inositol because of the groups connected. And lastly, we have diphosphatidylglycerol. It's called diphosphatidyl because di meaning two. We can see two phosphate groups here and then a glycerol. And the other name for this is cardiolipin. And they're saying phosphatidylcholine is the most abundant phospholipid. So that's the one that's found in the largest amount in mammal cell types. Okay, now they're just again going through the structures we saw in the previous slide. We have phosphatidylethynylamine. This is the ethynylamine, and another name for the whole thing is cephalin. It's found in nervous tissue of brain and spinal cord. And then phosphatidylcholine is also known as lecithin. It's found in foods like soybean and egg yolks. And it helps treat memory problems like in dementia and Alzheimer's. And yeah, and it's also the major component of the lung surfactant. So that's why lecithin is important. Now again, cardiolipin, the one we talked about, it's diphosphatidylglycerol. and it's the only dimeric phospholipid that we know. So it has 2 arms and it's dimeric. Cardiolipin is important because it's found in bacteria and eukaryocytes. In eukaryotes, it's only found on the inner mitochondrial membrane, and it's important for maintaining respiratory complexes. Cardiolipin is antigenic. So the body, if it goes outside, it can recognize it as not its own, and antibodies can attack it. So antibodies against Treponema pallidum, that is the bacteria that causes syphilis. So antibodies against it can also attack cardiolipin, and that can be a problem because we all have cardiolipin inside our bodies. So that would kind of antigenic. Okay. Normally antigens are, to put it simply, they can be called foreign proteins. But cardiolipin is not a foreign protein. It's something we have inside our bodies on the inner mitochondrial membrane. But the body can potentially recognize it as foreign and attack it. That's what I mean by antigenic. So if it had antibodies made against syphilis, those can attack the cardiolipin. And we're going to call those antibodies anti-cardiolipin antibodies. And if the body attacks itself, obviously it's going to cause problems. So this anti-cardiolipin antibodies can be seen in syphilis, like we said, because it's sort of the same antibodies. And then in a disease called antiphospholipid syndrome. So in this disease, you have a lot of thrombosis. Your blood just clots without any reason. And that would obviously lead to further problems. And then we have other diseases mentioned here. And antiphospholipid syndrome can be the cause of multiple spontaneous abortions. I'm not really sure if you need to know the diseases. Okay, now we're going to talk about something called plasmalogen. So we have the glycerophospholipid that we already had. And now at the carbon 1, instead of a fatty acid, we're going to replace it with an alkyl group. An alkyl group is basically a certain number of carbon and hydrogen. And it doesn't have any other functional group, so that's why it's just alkyl. And the way it's linked to the glycerol backbone is through an ether linkage. An ether linkage is the C connected to an O, and the O is connected to another carbon. So the COC bond is the ether linkage. So this molecule is what a plasmalogen is. And they're needed again for the membrane functions. Yeah, and for making lipid messengers, so things to send messages between different cells. Then we have something called platelet-activating factor. So this is an ether glycerophospholipid ether because it again has that ether linkage we talked about. But on carbon 1, the alkyl group it's connected to is saturated, because if we see here in a plasmalogen, it was unsaturated because it has this carbon-carbon double bond. But in platelet activating factor, it's saturated. There's no double bonds between carbons. So it would be that saturated alkyl group is connected to carbon 1. Carbon 2 is connected to an acetyl group instead of a fatty acid. And again, it has that phosphate because it's a phospholipid. It's made by many different cell types, and its function is to activate inflammatory cells. It causes hypersensitivity and inflammatory and anaphylactic reactions. So anaphylactic is it's what happens during allergies. It causes platelets to clump together and release the granules they had inside it. And also neutrophils and macrophages that are in the alveoli will make superoxide radicals. So basically it's just a It just causes an inflammatory reaction. Now we're talking about the synthesis of phosphoglycerides. So first we have something called cytidine diacylglycerol. So it's a glycerol with two fatty acids, that's why diacyl, and then this CDP is connected. Cytidine diphosphate, so that's the molecule connected to it, and it reacts with an alcohol. So after the reaction, this is what you get, the phospholipid. I'm not really sure how much details you're supposed to know about this reaction. And then the phospholipids are mostly made in the smooth endoplasmic reticulum. From there, they'll be taken to the Golgi apparatus, and then from there, they can go to the cell membrane, or they go out of the cell by exocytosis. And now we'll talk about sphingolipids. Like I mentioned before, the difference is that instead of the glycerol backbone, they have sphingosine as the backbone, and this is a molecule of sphingosine. And so we have something called sphingomyelin. So there we have sphingosine, and then it's connected to a fatty acid by an amide bond. So this one with the NH is the amide bond. And then it's also connected by an ester bond to phosphorylcholine. So this is the phosphate part, and this is choline. Sphingolipids are also important in the cell membrane. And again, like the glycerophospholipids, they have a hydrophilic part and two hydrophobic tails. And now for lung maturation. So when the baby is inside the mother, they don't really have to breathe. But after they come out, they have to. And so in between this time period, the lung has to mature. And we have something called surfactant on the inner side of the alveoli. It's made of protein and lipid, so it's a lipoprotein. And it's there to decrease surface tension, basically, because if the alveoli collapse, it's there to ensure that it can expand again and not have the walls stick to each other. So the surfactant we have is called dipalmitoyl phosphatidylcholine, also known as lecithin, and this is something we mentioned before. And the way to judge lung maturity is to measure the ratio of lecithin to sphingomyelin in the amniotic fluid. And if the ratio is more than 2, that says that the fetus has enough lung maturity. And it happens because the pneumocytes are making more lecithin than sphingomyelin, and this would happen around 32 weeks of gestation. And if there is an insufficient amount of surfactant, there would be something called respiratory distress syndrome. It can also happen, so for the fetus or the baby after it's born, it would happen if there was not enough lung maturity. But in adults, it can happen if the pneumocytes that make surfactants get destroyed by trauma or infection. And the way to treat it would be to give artificial, so synthetic or natural surfactants. And now I have some questions to kind of see if you got what we were talking about. So the first one is, what is the simplest phosphoglyceride? Can someone answer? Yes, the answer is C, phosphatidate. And the other 3 are just examples of phosphate-containing lipids. And yeah, and now this is the last question. What is the only known dimeric phospholipid? Yes, the answer is cardiolipin. So I think that's it for this presentation. Are there any questions? Here, yeah. All right, so if there are no questions, I guess that's it. Thank you for listening.